Welcome back. In this video, I will be discussing the molecular mechanism of the bacterial cell cycle, with my main focus on the associated proteins and their functions. The bacterial cell cycle encompasses the entirety of events from the birth of a new cell to its subsequent division, a phenomenon commonly referred to as binary fission. The binary fission process involves a series of sequential events. Initially, the cell undergoes elongation and chromosome replication. Then two newly formed DNA molecules separate and move towards the opposite pole, ensuring that each half of the cell contains one chromosome. Finally, a septum or crosswall is formed at midcell, dividing the parent cell into two progeny cells. While the bacterial cell cycle is notably less complex than its eukaryotic counterpart, but our comprehension on the underlying molecular events remains somewhat limited. Our current understanding is primarily gathered from the research conducted with model systems such as E. coli, Bacillus subtilis, the aquatic bacterium, Caulobacter crescentus. A significant breakthrough in understanding of bacterial cell division come from the identification of crucial genes through mutation studies. For instance, several temperature-sensitive mutant cells were isolated in E. coli that perform normal function at elevated temperature but fail to divide, resulting in the formation of a longer filamentous cell like this that eventually lies. The defective genes are called FtsZ. One such protein called FtsZ is conserved among all bacterial species, and its role is to form a central tightly packed spiral structure like this called the Z ring. A similar protein is also found in chloroplast and mitochondria. The Z ring marks the location of crosswall formation and eventually separation of the two daughter cells. It acts as a scaffold for the sequential assembly of the other proteins required for the cell division, collectively called the divisome. The FtsZ protein can assemble and deassembled in a GTP-dependent manner. For proper cell division, it is necessary to form the Z ring in the middle. Otherwise, unequal cell division may occur. Three other proteins called min C, min D, and min E inhibit the ring formation at the pole except during the sporulation process. Sim A only allows the ring formation where there is no or a very minimum amount of DNA is present, called the nucleoid exclusion. With the help of min C, D, and E and Sim A, Z ring formation is only possible in this region. When min C, D, E is mutated, the Z ring can form in this region, resulting in a mini cell without chromosome, which is deleterious. The Z ring also interacts with the inner lipid membrane with the help of GIP-A protein. Before the Z ring is formed, the microbial cell must complete chromosome replication. Bacterial cell contains a single circular chromosome and only a single origin of replication. All the machineries required for chromosome replication is assembled at the origin region, and the complex involved in the DNA replication process is called the replisome. From the origin, DNA polymerase initiates DNA replication and proceeds bidirectionally. At the TAR sequence, which is located at the opposite pole, replication is terminated. As you can see in this picture, as progeny chromosomes are synthesized, the two newly formed origins move towards opposite ends of the cell and rest of the chromosome follows in an orderly fashion. A protein called MARB, which is similar to eukaryotic actin, seems to involve in several processes, including determining the cell shape, chromosome movement, etc. The MARB polymerizes to form a spiral around the inside periphery of the cell, and the origin of each newly replicated chromosome associate with MARB, which then moves them to opposite pole of the cell. If MARB is mutated so that it can no longer hydrolyze ATP, its source of energy, chromosome fail to segregate properly. Not only that, if MARB is mutated, bacillus-shaped cell becomes spherical. The rod-shaped structure of bacillus is actually governed by MARB protein. Another protein called PAR proteins also helps in partitioning of the two origins to opposite cell poles and then physically pushing or pulling the two chromosomes apart. Upon completion of the chromosome replication and Z ring formation, the cell embarks on the final stage of the division, marked by the creation of a crosswall and cellular constriction. At first, autolysine hydrolyzed the beta-1,4-glycosidic bonds that connect NAG and NAM in the peptidoglycan backbone, which marked the site for the new cell wall synthesis. The divisome also contains enzymes for peptidoglycan synthesis such as FtsI, and FtsI is one of the several penicillin-binding proteins present in the cell. Penicillin-binding proteins are so named because their catalytic activities are inhibited by the treatment of antibiotic penicillin. Here, the old peptidoglycan chains serve as a primer for the synthesis of new peptidoglycan chains. As a result, a ridge is formed in this area, just like the barred scar in budding yeast. The FtsZ ring begins to depolymerize, triggering the inward growth of the wall materials to form the septum. This process generates a force that deform the membrane and initiate the constriction of the cell and seal off one daughter cells from other. You can read this portion from these two books, and for more you can also read this review article. So that's all for now. I hope you like this content. So share this with your fellow learners, subscribe and press the bell icon, and thanks for watching.